Thanks very much for the invitation. These are my disclosures. So I'd like to start by making the case for studying proteomics as a tool for investigating disease etiology. We're all familiar that common complex traits are a combination of genes and environment and the interactions of the two. Clearly, investigating epidemiologically environmental causes is tricky, proving causality is even harder. We've heard earlier how the genome offers very nice properties for causal inference, but a solely genetic approach does have its limitations, and these are that genetic associations do not always reveal the underlying mechanisms, and for common diseases, the genome is a poor therapeutic target. This might be different in rare Mendelian diseases, but generally for diseases like rheumatoid or diabetes, we wouldn't want to intervene directly on the genome. So a powerful approach is to study intermediate molecular risk factors that capture both the genetic and environmental effects. And it's now increasingly possible to measure these at epidemiological scale, and indeed not just one marker, but whole domains of intermediate molecules. And I think the study of proteins is particularly important because these are the effector molecules of biology, the functional units of biology, and also the target of most drugs. So from a translational point of view, insights from this kind of work is very valuable. So there are now a number of technologies for measuring the proteome at scale. These can broadly be divided into binding assays, like antibody-based techniques or aptamer-based techniques, or alternatively mass spec. Antibody techniques have been around for a long time, like ELISAs, but it's increasingly possible to do multiplex antibody-based assays. For example, the Olink platform that we've used. Olink offer panels that measure 92 proteins that are themed around various sort of concepts like inflammation or cardiovascular disease. And then the SomaLogic platform uses aptamers, which are modified oligonucleotides that combine particular conformations on proteins. And the latest iteration of the SomaLogic platform can measure about 5,000 proteins. So an early adoption of the SomaLogic platform was this study that looked at risk prediction in patients with stable coronary artery disease. And the authors of this study were able to identify a 9-protein signature that went on to predict cardiovascular endpoints or death. But of course, risk prediction is distinct from causal inference, and the main emphasis of our group is on the latter. So our approach to do this has been to combine genomic information with proteomic information. And the sort of core underpinning of this is so-called protein QTL mapping. So this is identifying the genetic influences on proteins, and in our case, we're studying plasma from the blood. And we use these really for two main applications. Firstly is to understand how disease-associated variants identified by GWAS influence disease. So filling in the blanks between genotype and clinical phenotype with techniques like co-localization. And the second application is this concept of Mendelian randomisation. So I'm not going to go into this in any great detail, but essentially the idea is to use genetics as a way of inferring from observational data whether an exposure is causal for disease. And what we're particularly interested in doing is using this kind of approach to anticipate the effect of targeting a particular protein with the drug and anticipate whether that's going to be effective before embarking on expensive randomized trials. So what we particularly want to do is create a so-called genetic dose-response curve where we have multiple genetic variants with differing effect sizes on the protein trait, and then we want to see how that correlates with disease risk. So this would be the sort of ideal situation where one might have common variants with small effect sizes and then some rare variants with large effect sizes, and we can read out how that translates into disease risk. So one of the bottlenecks that's limited the application of Mendelian randomization has been lack of suitable genetic instruments, i.e. lack of knowledge of the genetic determinants of plasma proteins. And to address this, our group established the interval study in collaboration with the NHS Blood and Transplant Service. And essentially this began as a pragmatic trial of how often it was safe to give blood, but as well as that, we've collected samples and created a multi-omic bioresource. So all 50,000 blood donors have been genotyped, and they also have various other layers of molecular data on top. And specifically with regard to proteomics, we've run the SomaLogic platform in around 3,000 individuals and 3 of the O-Link panels in around 5,000 individuals. And we've used this to perform a GWAS of plasma proteins using the SomaLogic platform. So this study came out last year. We discovered nearly 2,000 pQTLs, and this was an expansion on previous knowledge by approximately 4-fold. Here's a visual representation of our results. So on the x-axis, we have the genomic position of the pQTL genetic variant. On the y-axis, we have the genomic position of the gene encoding the protein. And what you see is this diagonal line colored in red here, which are local acting genetic variants that are influencing expression of proteins coded by nearby genes. So this is what we call cis-acting pQTLs. But in addition, you'll notice all the blue dots, which are genetic variants that are distant to the gene encoding the protein, often on a different chromosome, and these are so-called trans pQTLs. And in total, for the proteins that pass QC, we found a pQTL for around half. So I'm going to give you 2 examples of how this kind of knowledge can be applied for causal inference. So the first centers around MMP12 and coronary heart disease. So MMP12 is a matrix metalloprotease. It's involved in degradation of elastin and other extracellular tissues. There's been interest in MMP12 as a therapeutic target in COPD because in emphysema it's been linked to destruction of lung tissue. And there's also been observational associations of higher levels of MMP12 with incident coronary events. And that's led to speculation that MMP12 may play a role in coronary plaque destabilization. So we used our pQTL data to construct a genetic score for MMP12 levels. So we used 17 approximately independent genetic variants, and we correlated this genetic score against risk of disease. So what we found was that the genetic score associated with higher MMP12 levels actually was associated with reduced coronary disease risk. So this is in contrast to the observational data, and concordant with that also decreased stroke risk. And as a nice positive control, we found increased COPD risk. And when we sort of deconvoluted the variants that went into the score, you can see them here. Sorry, the error bars are not shown. I think that's a Mac to Windows issue. But essentially, genetic variants that increase MMP12 reduce coronary disease risk. So this raises the possibility that the observational data arises from reverse causation and that MMP12 may in fact be a protective compensatory effect against atherogenesis, and that this raises the possibility that therapeutically inhibiting it may be not such a good idea. So the second example concerns a rarer disease, ANCA-associated vasculitis. This is uncommon but a serious illness that's characterized by inflammation of blood vessels. It's an autoimmune disease, and the major subset have autoantibodies against a neutrophil protease called proteinase 3. And GWAS studies of this form of ANCA vasculitis have revealed an HLA signal, a signal near the gene that encodes PR3, and in the gene SERPINA1, which encodes alpha-1 antitrypsin, which is an antiprotease. So we looked at genetic determinants of the autoantigen PR3 in our healthy blood donor cohort, so this is not in patients with disease, and we found, using the O-Link assay and the SomaLogic platform, a very strong cis-pQTL, and this was consistent across the assays. There were 2 different aptamers that targeted PR3 on the SomaLogic platform. And here's a close-up of that signal, so you can see the peak signal lies just upstream of the PRTN3 gene. But interestingly, for one of the assays, this was one of the aptamers, there was a second signal that just met genome-wide significance in a missense variant in SERPINA1. And this is a well-characterized missense variant that results in intracellular accumulation of A1AT and defective secretion into the circulation, such that heterozygotes have around 50% circulating A1AT, and homozygotes have almost none. So this led us to speculate whether this aptamer was in fact binding a different form of PR3, perhaps bound to A1AT. And indeed, that's exactly the case. So we did pull-down assays in collaboration with SomaLogic. And what you can see here, so aptamer 2, when we do the pull-down, is binding to the PR3 in complex with A1AT, but hardly at all to free PR3. Aptamer 1 is binding both free PR3 and PR3 in complex. And importantly, neither aptamer binds A1AT in isolation, so this isn't a problem of non-specific cross-reactivity. So putting this all together, what this tells us is that the genetic signal influencing PR3 locally is increasing total PR3, and in fact there's an EQTL as well that's confirmed this, whereas the aptamer here is reflecting reduced abundance of alpha-1 antitrypsin. Therefore, there's reduced PR3 in complex with alpha-1 antitrypsin and increased availability of the free form of PR3. And so these two distinct genetic mechanisms lead to alter the exposure of PR3 to the immune system and presumably thus result in a loss of immune tolerance and the initiation of autoimmunity. So at the time, we thought our PQTL study was quite big, but clearly by GWAS standards, 3,000 to 5,000 individuals is not very large. So we are part of now the Scallop consortium, which is an international meta-analysis performing a protein QTL study using O-link assays. So Anders Malastig at the Karolinska is leading the analysis of cardiovascular panel 1. Jim Wilson's group in Edinburgh are leading a meta-analysis of cardiovascular panel 2 and 3. And in Cambridge, we're leading the inflammation panel analysis, and we have around 15,000 individuals from a variety of studies. And what we found from our inflammation analysis of the 91 proteins assayed, we found a PQTL for 71, and the ones where we don't find a PQTL, it's generally because the protein is not expressed at significant levels in blood. And with this increased power, we're seeing more and more trans PQTLs. So this is just an example of multiple trans signals for the protein IL-12B, as well as this very, very strong cis signal. But interestingly, the Manhattan plots do look somewhat different to Manhattan plots for a similar size of study if one was, say, looking at BMI or height. And actually, most of the Manhattan plots, there's 2 or 3 signals. So I've put question mark here because clearly this is only looking at the genome-wide significant hits. But presumably, because proteins are under much more proximal control of DNA, we're seeing fewer influences than we would on a more downstream phenotype like, say, BMI. And here's the distribution of number of regions associated according to number of proteins. So as a penultimate slide, I think the time has now arrived where omic technologies, which were previously used to measure lots of things in small numbers, can now be scaled up to epidemiological scale. I hope I've convinced you there's value for etiological inference, for therapeutic target prioritization, touch briefly on prediction, and the other area I didn't touch on but I think is important is using clustering approaches to pull out molecularly distinct subsets within one particular disease and then correlate those things with different outcomes or responses to treatment. I should finish up on a word of warning. The protein universe is complex. I've already alluded to the problems of free versus complex proteins. There's also the issue of post-translational modifications, alternate isoforms. Pre-analytical variation is always an important source of difficulty when you're doing really big epidemiological studies. And the binding assays I've mentioned are vulnerable to a number of issues. One is binding the wrong thing, so off-target cross-reactivity, and also if there's a protein coding variant that changes epitope shape, it can alter the binding of the assay and produce an artifactual PQTL. So on that note, I'd like to thank all my colleagues and collaborators, and thanks very much.